This recording is the somatic nervous system, part 2 of 4, hearing and equilibrium. In understanding how hearing works, let's look at the structures of the ear first, beginning with the largest portion of the external ear, which is the auricle or pinna. That is a funnel-shaped object that helps direct sound waves into the external acoustic or external auditory meatus. The helix is the outer ridge of the auricle or pinna, and inferiorly you will see a lobule that is mostly adipose connective tissue. The external ear continues internally until the tympanic membrane is reached. The tympanic membrane is a semi-translucent membrane that's roughly the diameter of an M&M. Most people know the tympanic membrane as the eardrum. However, since you are in anatomy and physiology, you're going to be calling it the tympanic membrane. Attached directly to the tympanic membrane is the first of 3 auditory ossicles, which is the malleus. The malleus directly attaches to the tympanic membrane, and the malleus forms an articulation with the incus, which in turn forms an articulation with the stapes. The malleus, incus, and stapes are the 3 auditory ossicles. The stapes communicates with a membrane that is shaped like an oval window and hence is named the oval window. You'll notice that inferior to where the stapes communicates with the oval window, there is another structure that has the name of the round window. This is also an opening that has a membrane covering over it. And what happens is when sound vibrations start causing the tympanic membrane to vibrate, those vibrations are transferred through the auditory ossicles, amplifying them, and the stapes is going to push fluid that is on the opposite side of the membrane for the oval window. And as fluid gets pushed through, it travels through this snail-shaped structure called the cochlea, which is divided into different regions. And after the fluid reaches the apex, it will continue to flow in the opposite direction in another chamber, all the way back to the round window, which will in turn vibrate because of the vibrations that are being pushed on the oval window by the stapes. You'll notice that there is also another structure besides the cochlea, which is the vestibule and the semicircular canals. So the cochlea serves as a structure that's involved in hearing, whereas the vestibule and semicircular canals are going to function for equilibrium or balance. The external ear, you can see at the very top picture, includes not only the external structures but also the external acoustic or external auditory meatus up until the tympanic membrane. The middle ear includes the tympanic membrane and the auditory ossicles and this middle ear cavity. And then the inner ear or internal ear is often referred to as a bony labyrinth because of the cochlea and the vestibule and the semicircular canals. You'll also notice a structure inferior to the vestibule, semicircular canals, and cochlea, which is the pharyngotympanic or auditory tube, which goes from the middle ear cavity and goes all the way to the pharynx. And there is a flap of membrane that covers over the tube where it enters the pharynx. So when people go up in altitude, what happens is the air pressure decreases as we go higher in altitude, and if air pressure decreases on the outside of the tympanic membrane, then we have to equalize pressure by decreasing pressure in the pharyngotympanic or auditory tube so that the pressure on both sides of the tympanic membrane is equal. If you ever go up in an airplane and you notice people start yawning as the airplane ascends, that opens up the membrane flap to the pharyngotympanic tube, thereby allowing pressure to escape and decrease, equalizing pressure on both sides of the tympanic membrane. If pressure cannot be equalized, there are a lot of nerve endings that go to the tympanic membrane, and that can cause a lot of pain. You'll notice that there is a vestibular nerve coming off of the vestibule, as well as a cochlear nerve, which arises from the cochlea. These two nerves join together to form cranial nerve 8, which is the vestibulocochlear nerve. Here again is a picture of the external ear, which functions to collect and transmit sound waves to the tympanic membrane, and the middle ear where the auditory ossicles are. The auricle itself, or pinna, is mostly made up of cartilage and epithelial tissue. The external auditory meatus, or external acoustic meatus, is the canal through which sound will travel, and you've already seen that opening in the temporal bone. This canal is lined with ceruminous glands, which secrete earwax, otherwise known as cerumen. Cerumen helps to trap foreign matters, and it also has antibiotic properties to help keep the ears clean. The tympanic membrane is the boundary of the external ear. It's made up of epithelial tissue and vibrates when sound waves hit it. This takes us to the middle ear, which starts at the tympanic membrane and includes the 3 auditory ossicles, the malleus, which directly attaches to the tympanic membrane, the incus and the stapes. These 3 auditory ossicles serve to amplify sound waves. We also see the opening for the auditory tube or pharyngotympanic tube. This is also referred to as the eustachian tube, if you've ever heard of that term before, and it helps us equalize pressure on both sides of the tympanic membrane. The auditory tube / eustachian tube / pharyngotympanic tube helps connect the middle ear cavity to the throat, specifically the nasopharynx. There are 3 portions of the pharynx that you'll learn about when you get to A&P 2. There's the nasopharynx, the oropharynx, and the laryngopharynx. So the nasopharynx is the uppermost portion, and the auditory or eustachian tube or pharyngotympanic tube connects the middle ear to the throat. There are special muscles associated with some of these structures to limit vibrations, thereby minimizing damage that could potentially occur, including the tensor tympani muscle that you can see here. And there's also a little stapedius muscle. Again, if there's a really, really loud sound, these muscles can contract to help limit the vibrations, thereby helping to minimize the damage that may occur. Otitis media is a condition where the middle ear cavity has become infected and inflamed, and it's usually the result of a sore throat. Sometimes when people get strep throat, the infection can climb up the eustachian tube to the middle ear, and that will cause the tympanic membrane to become very red and swollen and bulgy, like you can see here in the upper picture. This is actually one of the most frequent causes of hearing loss in children, and again, if it's acute, it will cause the tympanic membrane to bulge and become very red and angry in appearance. It's most commonly treated with antibiotics. Most kids with ear infections usually get amoxicillin, and sometimes if it's really bad and doesn't go away, they might give them banana-flavored Augmentin. If pus or fluid accumulates, then pediatricians may decide to go ahead and have the child get drainage tubes inserted into the eardrum. And these tubes do damage the tympanic membrane, and they often will fall out. It does temporarily relieve the situation. However, most children get ear infections because when kids are little, you'll notice that their heads are very, very round. They elongate as we grow older, and because of the orientation of the eustachian tubes, with the head being rounder, they're more horizontal. And when a baby is delivered, the doctor will often use the head as a lever to get the baby out of the birth canal, and that can displace atlas and occiput. And if atlas becomes displaced, that can actually cause the eustachian tube to become blocked. So sometimes it's as simple as getting a child adjusted to stop the ear infections from occurring, and it's usually very, very successful in terms of the rates of success for ear infections in children. When we look at the inner ear, we know that hearing occurs in that snail shell shaped structure called the cochlea. It's actually about the size of a split pea, and the cochlea includes 3 tubes that are lined by membranes inside what's called a bony labyrinth. And a labyrinth is just a fancy word for a maze. The 2 outer tubes are the scala vestibuli, or vestibular canal, and the scala tympani, which forms the tympanic canal. And then there's a middle canal or inner canal that is referred to as the cochlear duct. And this cochlear duct is filled with fluid called endolymph, because endo means inside, whereas the scala vestibuli, or vestibular canal, and the scala tympani, or tympanic canal, are each filled with fluid known as perilymph. Now, the vestibular canal and tympanic canal will actually communicate with one another at the very apex of the cochlea, which is called the helicotrema. So we'll see that in just a second. The production of sound occurs because of this special mechanism called the organ of Corti. And we'll take a look at the organ of Corti and the structures that produce it so you can understand how sound is produced a little bit better. So here, if we look at the scala vestibuli and scala tympani down below, we see the cochlear duct in between, which is sometimes called the scala media, and we have a blown up reproduction of the organ of Corti. You'll notice that there is a membrane at the base of the organ of Corti called the basilar membrane, because it's on the base. This is at the upper portion of the scala tympani. There's also another membrane associated with the organ of Corti called the tectorial membrane. And as you look at the cells involved, we see outer hair cells, and then we see inner hair cells. Now, it's actually a concerted effort between the hair cells that produces sound. We also have supporting cells that help support the hair cells. The hair cells have tiny little hairs called stereocilia that project up into the tectorial membrane, so they're actually kind of embedded in it. And here we can kind of see at the picture below the outer hair cells projecting through the tectorial membrane and the inner hair cells doing the same. Now, what happens is when fluid flows through the scala vestibuli, the waves of fluid actually displace the membrane of the scala vestibuli, which causes displacement of the scala tympani below it. That moves the basilar membrane. And the outer hair cells have the job of fine-tuning the basilar membrane, because it's really the inner hair cells that are going to produce sound, and the outer hair cells may either amplify or dampen the actions of the basilar membrane, and that's what affects the inner hair cells to produce sound. So you'll notice that the afferent sensory fibers are attached to the inner hair cells, whereas the outer hair cells actually have efferent fibers going to them to, again, kind of change how the basilar membrane responds to the vibrations. Here we have a bigger picture of the organ of Corti, where we can see the outer hair cells with their tiny little hairs called stereocilia embedded in the membrane above called the tectorial membrane, which does not make contact with the scala vestibuli, by the way. It's separate and detached from that. We have the inner hair cells and afferent nerve fibers coming from the inner hair cells. The job of the outer hair cells is basically to either amplify or dampen the effects of the basilar membrane, because what happens is when fluid vibrations start going through the scala vestibuli, it displaces the basilar membrane associated with the scala tympani. And because it's the basilar membrane on the scala tympani that's going to be moving, that causes the hairs embedded in the tectorial membrane to move. And when those stereocilia move, the pressure wave will bend those tiny little stereocilia, allowing potassium channels to open. When potassium comes in, that initiates an action potential depolarizing the membrane of these hair cells. And then when the membrane depolarizes, that causes an influx of calcium to come in, because those are voltage-gated calcium channels. And then when calcium comes in, that causes a release of a neurotransmitter into the synapse between the hair cells and the afferent nerve fibers. So that's basically how sound is produced. It's really from the inner hair cells and the afferent nerve fibers. However, the outer hair cells play a really big job in either amplifying or dampening the effects of the basilar membrane. So in terms of sound frequency, we can hear sound frequency usually between frequency of 20 hertz and 20,000 hertz. If we unrolled the cochlea, we would see the scala vestibuli next to the base of the oval window, and the scala tympani that has the basilar membrane associated with it, that's going to be on the lower aspect and communicating eventually with the round window. Now, sound waves of a higher frequency are actually going to be heard closer to the base of the cochlea, where sound waves of a lower frequency are going to be able to travel to the apex of the cochlea. And remember, these two are actually going to communicate with one another at the apex. Here we can kind of see the displacement of the membrane of the scala vestibuli, which ends up displacing the basilar membrane, which is attached to the scala tympani. And it's that displacement that causes the tiny little stereocilia, which are embedded in the tectorial membrane, to bend. And remember, when they bend, potassium goes in, depolarizes the membrane of the hair cell. That depolarization opens up calcium voltage-gated channels, which triggers calcium to come in, which triggers the neurotransmitter in the hair cell to be released to the afferent branches of the cochlear nerve. Here we can see the stapes vibrating against the oval window of the scala vestibuli. As these vibrations go through, you can see the displacement of the basilar membrane that is attached to the scala tympani, right here. And again, the stereocilia embedded in the tectorial membrane are going to bend. So if we were looking at this from the very beginning, we'd look at sound waves coming in to the external acoustic meatus, vibrating against the tympanic membrane, having those vibrations amplified by the malleus, incus, and stapes. The stapes then vibrates against the oval window, which causes the perilymph in the scala vestibuli to vibrate, displacing the basilar membrane, which is associated with the scala tympani. And then as that fluid continues to vibrate all the way around the helicotrema and the apex, coming around the scala tympani, it's going to vibrate against the round window. So the round window is basically just going to vibrate because it's a membrane. It's kind of like a little elastic membrane that will bulge and contract with the vibrations. So again, as the stereocilia are displaced, those afferent impulses go and get transmitted to the cochlear nerve, and the cochlear nerve joins with the vestibular nerve to form the vestibulocochlear nerve. Here we can see the pathway of hearing as those afferent neurons from the inner hair cells travel to the medulla oblongata, and what happens is eventually neurons will take this incoming afferent sensory information to the thalamus, and then from the thalamus to the primary auditory cortex in the temporal lobe. I could explain every single little bit of this pathway and you'd feel compelled to memorize it, and I don't want you to feel like you have to memorize it, so I'm not going to do that to you. Understand that with all sensations, whether they're taste or auditory or visual, the primary cortex for any sensation is just going to give us information about a sensation. It is the association cortices in the cortex that will give us meaning. So the auditory association cortex will give us meaning to whatever sound we have a sensation about. So if crying is the sensation that we hear, then the association cortex, the auditory association cortex, will give us meaning to the type of crying that we're hearing. Is it crying that a baby's crying because it's hungry or needs to be changed? Is it crying that somebody's happy or crying because someone's hurt? And so these association areas are really important because we use those to determine what the sensations are, and that's we perceive things in the association areas. This takes us to equilibrium. We know that hearing occurs in the cochlea. I don't know if I remember telling you this, but the cochlea is about the size of a split pea. That's how tiny this is. Equilibrium is established by two other areas in the internal ear, including the vestibule and the semicircular canals. Both the vestibule and the semicircular canals are lined with membranes, just like the cochlea was lined with membranes. When we look at the vestibule, we can see that there are particular areas within the vestibule, including an area known as the utricle and an area known as the saccule. Now, the utricle and the saccule make up structures collectively known as maculae. The word maculae means spots. The utricle is responsible for our understanding of linear acceleration on a horizontal plane, including tilting the head, whereas the saccule helps us determine linear acceleration vertically, such as if you were in an elevator. So those areas, the utricle and the saccule, again, are located in the vestibule. This area is the utricle. You can see that purple spot, and this area is the saccule. You can see that little purple spot too. The semicircular canals consist of 3 different canals oriented in different directions, and we have anterior, posterior, and lateral semicircular canals, and there are special structures in the ends of the semicircular canals that are referred to as the cristae ampullaris. The cristae ampullaris help us understand rotational acceleration on 3 different planes. Static equilibrium is determined by the maculae found within the utricle and the saccule in the vestibule. And the utricles help us understand horizontal linear movements and bending the head forward, where the saccules help us understand vertical acceleration and deceleration. And these portions of the vestibule help us maintain equilibrium whenever our head position changes. So any kind of gravitational perception, linear acceleration or deceleration is going to occur because of these particular structures within the vestibule. Again, the utricle and the saccule collectively are called the maculae. So what happens is within the utricle and the saccule, there are, again, hair cells. And the hair cells have a gel-like structure over them with tiny little ear stones called otoliths. And what happens is, if I bend my head forward slightly, the otoliths and gravity are going to move the gel, causing the hairs to bend, and that's what causes an action potential to occur to give my head the understanding and my brain the understanding of where my head is in space. When we look at the semicircular canals, these also help detect head movement, anything that's rotational, like saying no, or side to side, kind of shoulder to shoulder movements, or even forward and backward when it's nodding like this, as opposed to just tilting the head. If it's going back and forth, then that's more motion that's picked up by the semicircular canals. Static equilibrium occurs when our body is in one position, but we may be moving, such as driving in a car, linear acceleration. Our body is not changing position, but we're being moved forward by the car, or the same thing going up and down in an elevator. Or if we're sitting, we change the position of our head. That's all static equilibrium. However, dynamic equilibrium is what occurs when our body is moving. So you see this with gymnasts a lot, or even somebody walking, any kind of body motion when we move our bodies and our heads at the same time. If you've ever seen a dancer or a figure skater twirl around and she's spotting on a particular point, that's a process that helps people maintain equilibrium. So rotational acceleration, usually when the body's turning or moving, this is going to play a big part in dynamic equilibrium. And this is done by the Christi ambularis in the very beginning of the semicircular canals, where they join together with the vestibule. There is a structure, part of the Christi ambularis is the swollen portion of that semicircular canal that's referred to as the ampulla. The semicircular canals and ampullae are filled with endolymph, they're filled with fluid, and there is a tiny little ampullary nerve in the center and the base of the ampulla that is going to receive signals from, again, hair cells. These hair cells have a gel-like structure called a copula that is situated on top of them, and so the stereocilia of the hairs are embedded in this gel-like copula. And when the head rotates, the fluid moves, bending the copula in the direction opposite the rotation. And again, it's just the bending of the hair cells, of the hairs in the hair cells, that will trigger action potentials to cause us to understand where we are in space during dynamic equilibrium. So there's a good video on this. You can watch if you like. You can't get it on this particular video because the links don't work on these videos. I don't believe they do. However, there is a regular PowerPoint that you can use to check out any of the links that I've got on these for your viewing pleasure. And they're going to go into more depth. For these things, what I really want you to know more about is that the utricle and saccule are part of the vestibule as well as the semicircular canals. The semicircular canals are where we find the Christi ambularis, and you should know what portions are for dynamic versus static equilibrium, and linear acceleration versus vertical acceleration or deceleration. And then know about hearing that we discussed a little bit earlier. People often ask about motion sickness, and motion sickness often occurs when sensory input is mismatched. For example, if a person's in a car and the visual input indicates that that person is in a fixed stationary environment, but the vestibular apparatus detects motion that disagrees with the visual information, the brain receives those conflicting signals, and then the person may start to salivate and sweat, and that could lead to vomiting or nausea. And usually different types of over-the-counter drugs can depress the vestibular inputs, and it's always best to do this before a person gets into the car. Seasickness is a really good example of motion sickness, because with seasickness, the motion of the ocean is moving the boat up and down, and the horizon's not moving, so the person gets this feeling of moving up and down, whereas the horizon is steady and stable. So that's another good example of motion sickness. So that ends part 2 of this particular chapter.